Hello again. Welcome back to the Classics of Immunology Journal Club. And before we get started, I just want to remind you to help out by liking the video and clicking on the bell and subscribe to the YouTube site and scroll down on the YouTube site and you'll find all my contacts for my website and social media. And if you go on my website, you can find all the classic papers in immunology that have been posted there, including the one I'm going to talk about today, which is from a Journal of Immunology in 1998. So we're getting closer to the present time. This paper is by Andreas Cerutti, Hong Zhan, Andras Schaffer, Leif Bergsagel, a couple of other people that I don't know, and Paolo Casali, who is the senior author. And Paolo and I are old buddies from Cornell. And so this is a very interesting paper and really one of the classics in immunology. And the title is CD40 ligand and appropriate cytokines induce switching to IgG, IgA, IgE, and coordinated germinal center and plasmacytoid phenotypic differentiation in a human monoclonal IgM+, IgD+ B cell line. And that's a long way of saying this is all about T cell health. Now, you may recall, in terms of the background on this kind of thing, when we first cloned cytotoxic T cells back in 1979, and when we published that paper in the introduction and or the discussion, I said, the ability to work with T cell clones rather than heterogeneous populations of cells is going to allow us to find the molecular mechanisms of all kinds of things that everybody wants to know about, including the molecular nature of the T cell antigen receptor, the molecular mechanisms responsible for T cell killing, cytolysis, as well as the molecular mechanisms responsible for T cell help. And of course, the T cell receptor was worked out by Alice Reinherz soon thereafter in a series of papers in 1983 that we reviewed. And then throughout the 80s, a couple of investigators really solved the problem of how T cells kill other cells. T cell mediated cytolysis. Eckhart Podak, Pierre Henkart were instrumental in those experiments and discoveries. But B cell help, T cell help for B cells. And, of course, because I'm a T cell person, I always give them a little dig saying that, that B cells were sort of stupid. They didn't know what to do. They needed to have T cells tell them what to do. And well, that took a while to work out what was really going on in terms of this T cell help for B cells. The phenomenon goes back to the 60s and 70s when Mitchison and others, and we reviewed those papers in this journal club, and you can go back and watch those videos too, showed that there was something funny going on in terms of what they called cognate recognition. In other words, the T cells and the B cells had to have very intimate contact with one another. They had to really sort of kiss one another in order to get the whole game started. Ultimately, that was solved by the end of the 80s, early 90s, and by the discovery that there was a cell surface receptor called CD40 on B cells, activated B cells. That receptor would be triggered by a molecule that appeared on the surface of T cells, activated T cells. That was then labeled CD40 ligand. And back in 1991, a series of papers came out by several different laboratories showing that, or in the 80s, 89 and 90, showing that this trigger from CD40 ligand to CD40 was really critical for making the B cells proliferate and differentiate and really do their thing. And Jacques Bonchereau had a paper out, I think it was in Science, that we also reviewed here in 1991, showing that if you used antibodies to CD40 together with interleukin-4, you could get long-term B cell proliferation. And out of that discovery, I was sort of curious. I thought, well, this is going to be the crack in the B cell whole alliance. because if you can get long-term B cell lines, you got to be able to clone those long-term B cell lines, just like we did with cytotoxic T cells, and then others did with helper T cells. And then you'd have the cells to be able to study this whole thing in terms of what the B cells are doing when they were getting help from the B cells, and exactly what kinds of help CD40 was giving, CD40 ligand, and interleukin-4 and all the other interleukins. And we went through those in the Journal Club as well. Well, in the background of this paper, Andrea Cerutti, who was the first author, was a postdoc from Italy who got together with Paolo Casali at Cornell. And this paper is the result of that. And this paper was published in 1998. So that's basically a decade from the discovery of the importance of CD40 and CD40 ligand in activating the whole B cell program. And then Paolo Casali I first met at Cornell when I went there in 1993. He came there soon thereafter. I think it was like 95, 96 or so. Anyway, we got together and we had a journal club together, and that was a lot of fun. And I learned a little bit about B cells, but I have to tell you, these B cells are very complicated. They're not as simple as the T cells. And this paper is one of the things that lets you know that. So in the introduction of this paper, they go through and they sort of review the whole story about B cell maturation and differentiation. They don't really delineate these two processes. And I found, as a hematologist first in my clinical years, I was well schooled that whatever happened in the bone marrow in terms of hematopoiesis, that was maturation of whatever series you're talking about. Let's say, look at red cells. Red cells matured in the bone marrow from red cell precursors, erythroid precursors that did not make hemoglobin, to the point where they ultimately gradually made hemoglobin over time in the bone marrow until they ultimately extruded their nucleus, and they end up being a big sack, a big bag of hemoglobin, which then, when the cell was mature, then it would leave the bone marrow and go out into the blood and into the periphery and function. Immunologists, they really sort of miscegenated maturation and differentiation, but this paper doesn't. And this paper does is they pointed out in the introduction that B cell development takes place in two different phases. One is antigen independent and occurs in the bone marrow. And in that situation, a precursor of a new B cell, a mature B cell, ultimately is a cell that will become a mature B cell, matures and expresses ultimately on the cell surface IgM and IgD. That's maturation. That occurs in the bone marrow. And once that cell becomes mature, leaves the bone marrow, goes out into the periphery, populates the lymph nodes, the spleen, and the peripheral blood. Then there is a process that's called antigen dependent, that it was termed or found to be antigen dependent. And that causes the antigen activation of the B cell, initiates a program that ultimately determines what kind of a B cell this beast is going to become. That's differentiation. And the end result of B cell differentiation is the generation of a plasma cell that is especially geared up to crank out huge amounts of antibody, immunoglobulin molecules into the periphery. The other fate of a differentiated B cell is a long-term memory B cell that doesn't secrete huge amounts of antibody, but it has that capacity should they see antigen for the second time. In order for differentiation to occur, there are two processes that had been worked out in the B cell by the B cell people over this time span from, like, say, 1960 to 2000, essentially. This is the time span we're talking about. One of these processes is called heavy chain class switching, where at the genetic level, heavy chain is determined by a process of VDJ recombination, so that the genes are put into close proximity to one another to make a mature immunoglobulin molecule. And the heavy chain class switching then determines the isotype of the antibody, the ultimate gene product of this whole process. And the isotype, whether it's IgG and 1, 2, 3, or 4, or IgA and be a secretory immunoglobulin, or IgE and be an allergenic immunoglobulin, that determines the effector capacity of an antibody in the B cell, essentially. And that's very, very important in terms of the generation of an immune response that's effective in fighting off against all these microbes that we've got out there. The other process that occurs in the introduction, or that they review in the introduction, is affinity maturation, or somatic hypermutation. And so that in that process, there is an ongoing random somatic mutation of the antigen binding region of the antibody that ultimately determines the highest affinity, or results in different affinity antibodies that are then selected out by the antigen, and particularly as the antigen concentration diminishes over time, because it's being removed by the body, you ultimately end up triggering the highest affinity antibody producing cells as a consequence of hypermutation, somatic hypermutation. And that is another process that is instrumental in terms of making the B cell immune response today what it is. Both of these processes of class switch recombination and somatic hypermutation occur in a sequential fashion over the course of the differentiation of the B cell from being just a surface IgM positive, IgD positive cell, to ultimately taking on the morphology as well as the functionality of a plasma cell or a memory B cell. The problem that was confronting all of the investigators in the B cell compartment, I alluded to earlier, and that was they were, because of the fact that they did not have long-term B cell lines, antigen specific, and then also frozen in a state of, at the end of maturation from the bone marrow, so that they were just beginning to be B cells. They'd been matured, but now they had to differentiate. They didn't have those things, and so ultimately, what Paolo did, and Andrea and all the rest of them in the lab, is that they searched through. There were all kinds of long-term human B cell lines. And of course, in immunology, if you weren't a mouse immunologist, you weren't really a real immunologist. You had to work on mouse cells to be in the right club, in this whole right clique in this whole process. But Paolo, he focused on human things, and he never varied from that, as far as I know. Maybe he could tell me whether or not he did. The thing is, they went through a whole cadre of long-term B cell lines, looking for ones that would have just a mature phenotype, surface IgM positive and IgD positive, but not anything else. And they found one, and in terms of a cell line that we'll get into in terms of the materials and methods. And that's what they're going to focus on in this paper. That was critical, because what they found then, and what they summarized at the end of their introduction, is that they found a common differentiative program that involved triggers from T cells, CD40 ligand, interleukin 4, interleukin 10, which is actually produced by B cells, and interleukin 6. And so all of those cytokines, in addition to the CD40 ligand, were really, that's what T cell help is. So that's why I selected this paper. Now, in the materials and methods, They talk about the cells and how they came to this B cell line that they used for this paper. They tested over 25 human B cell lines for switching of surface IgM to IgG under the influence of CD40 ligand and IL4 or IL10. They just threw that in, and they were looking to see if they could switch from M to G because they wanted to study that. They found one particular cell line that's called BL16. That's its name, and it was isolated from a patient with Burkitt's lymphoma. Burkitt's lymphoma was a very rapidly growing and fatal lymphoma, usually the onset in children. And so this particular B cell line was surface IgM positive and IgD positive, but that was it. And so Paolo and the rest of them cloned this cell line and selected out for the highest producing cells with the highest frequency of switching from IgM-D to IgG. Then the rest of their materials and methods were pretty straightforward. They measured immunoglobulin secretion and cytokine secretion with ELISAs that were available. And then they used flow cytometry and fluorescence microscopy and electron microscopy to look at the B cells under different kinds of situations. We should go to the results. And I'm not going to show you all the results. There's a lot of results in this paper. And if you happen to be a B cell guru and you want to really know the nitty gritty, you should go and read this paper carefully and look at all the results and to see exactly what you think of them. So this is figure 2 from this paper. And there's an upper panel and a lower panel. And this upper panel, they just simply called these cells clone 1. The clone 1 cells at time 0, and then at day 5 in culture, and then day 8 in culture. And of course, the day 0, this is the phenotype of the cell. So this is IgM, surface IgM, and then surface IgG, IgD, IgG, IgA, and IgE. So the cells are basically double positive for M and D, and they are G negative, they are A negative, and they're E negative. So then after 5 days of culture with CD40 ligand and interleukin 4, you can see that now we've gone from 99% double positive to only 50% IgD, IgM double positive. And we've got down here 4% and so forth in terms of looking at so they lost some IgM and in IgD, and they've gained IgG. And here we've got about a total of 15% positive with IgG. And they've also gained some IgA positive cells and some IgE positive cells. That's day 5. And on day 8, you can see that it's you even get more now of about the same of IgD, but then more IgG positive, IgA positive, and IgM negative cells. So that's that. And then on the lower panel here is actual secretion into the supernatant of the different immunoglobulins. And so this is after culture of negative or nothing, and this is IgM. Here is the negative control for CD40 ligand. They used CD8, and again, it's IgM positive, and that's it. And then when you use CD40 ligand, now you've got IgG and IgA. And here, if you then add in an antibody to interleukin 10 or to TGF beta, which they surmise are being made by the clone 1 cells themselves, you can ameliorate this differentiative switch. If you add interleukin 4 into that whole mix, now in addition to G and A, you also get IgE positive cells here. Then moving on, if you go on to figure 4 is where I want to go next. And this is figure 4. That's figure 5. Here's figure 4. The germline and VDJ immunoglobulin recombined transcripts induced by clone 1 cells. So what we have here in the upper panel is, or what we're stimulating with here is then the CD40 control, negative control, the negative control plus interleukin 4, and then the CD40 ligand alone, and then the CD40 ligand plus interleukin 4. And this basically is the essence of the paper. You can see what's going on by these experiments here. So that in A and E, we've got the negative control, A here and E here. There are no germline transcripts. And here, if we add interleukin 4, there's no germline transcripts. And if we look at then C, where we've got CD40 ligand alone, we have, for the first time, germline transcripts appearing in here and we see transcripts for gamma 1, gamma 2, gamma 3, and alpha 1. And when we look at adding interleukin 4 into this mix, we have all of the germline transcripts and the recombined VDJ recombined transcripts here. So that's all I want to really go into in this particular thing, because as I said, the rest of the data you should look at very, very carefully. This is for the differentiation of different markers during this whole process under these stimuli. And down here, you see what the cells look like by electron microscopy. This is the time 0 cells. These are the ones that have had all of the cytokines plus interleukin 6. And here, for the first time, you see large nucleoli, because they're cranking out messenger RNA, and they've got very convoluted or enhanced endoplasmic reticulum, because they're making immunoglobulin molecules, essentially. So we should stop the screen sharing at that point and go into the discussion of this paper. So the very first statement they make in their discussion is that the CD40 ligand plus interleukin 4 plus interleukin 10, because they added interleukin 10 in some of these experiments. They got even more of whatever they got without it, adding it. It induces this human clone 1 BL16 cells to undergo switch heavy chain recombination to all of the isotypes. Now, all of the IgGs, IgA, IgA and IgE. If you add that in interleukin 6, then you get plasma cell differentiation. So, of all of the cytokines, particularly the interleukins that we've talked about, that happened over this 20 years between at the end of 70s to the end of the 90s, interleukin 4 alone, without CD40 ligand, was insufficient to induce class switching, heavy chain class switching. And you needed both interleukin 10 and TGF-beta produced by the normal activated B cells, or this clone 1 cell line, in order to get a progression through the different stages of B cell maturation or differentiation, including centroblastic, centrocytic, and memory differentiative stages. And finally, it was clear in all of these experiments that the role of interleukin 6 was to cause differentiation to a high immunoglobulin producing plasma cell. So that's heavy chain class switching. And the other process that B cells undergo is, as I said in the introduction, is the somatic hypermutation, which accounts for affinity maturation. And that is activated by the surface immunoglobulin. And they don't go into that particular process, except for the fact that they say that if you activate the surface immunoglobulin of the clone 1 cell lines, that they will then undergo affinity maturation. So stay tuned. There's more to come in terms of this T cell help for B cells. And don't forget to like the video and click on the bell, subscribe. You'll get more. And check out the website. The other thing is, well, I'll leave that there. So thanks for listening in, and come back next week.